Mutations, a change in our DNA. Whether they're caused by radioactive materials, X-rays, or smoking, they can cause cell changes that could lead to cancer or uncontrolled cell division. This lesson is on mutations and other issues that happen with transcription and translation. So a mutation is a permanent change in the DNA sequence, and it can happen randomly, or it can be caused by mutagens or mutagenic agents. Mutations can be inheritable if they happen in the germline, so where the sperm and eggs are produced. And they can arise from mistakes in DNA replication when one DNA base, nitrogen base, is substituted for another. And then this creates new genetic code. Now, this could make a new amino acid, which causes a different protein to be made. So mutagenic agents can be physical, where something has been pushing or tugging on chromosomes, causing a change. It can be chemical, such as carcinogens, mustard gas, or even poor nutrition. Some antibiotics are mutagenic. Radiation, so X-rays, so if you're at the dentist, how you often have that lead vest to wear if they're X-raying your head. Ultraviolet radiation from the sun and cosmic rays. There can be natural mistakes made during mitosis or meiosis. And nutritional, when we say lack of certain nucleotides in the diet, means we're unable to provide the proper free bases, and this could cause a mismatch as the cell is just trying to do the best it can. And there can be biological mutagenic agents. So most viruses use genetic material of chromosomes to reproduce, and they join existing DNA and might cause permanent damage. So if a mutagen is present in the gametes, the sperm or the eggs, it will be passed on to further generations. And this is why it's particularly dangerous for pregnant women to come across mutagens. So X-rays, certain antibiotics, and chemicals, because as the gametes are developing in her ovaries to maybe possibly make eggs for the next generation, there can be issues if men have radiation near their testes. As sperm are being made, there can be mutations there, or as she's growing the testes or ovaries of her unborn child, there can be mutations in those cells. Mutations are generally now grouped under two categories. We have chromosomal mutations, which are large, and point mutations, which are in the nitrogenous base area. So large mutations visibly affect the structure or number of chromosomes. So non-disjunction, where we might have trisomy or monosomy, or things like Fragile X syndrome, where the X chromosome is damaged. Point mutations alter a single gene, and there are several different types. We have base substitution, where one base is substituted into the DNA strand for another base, which could result in one amino acid being affected. So ACG-CCA could become CCG-CCA. And for example, in sickle cell anemia, the substitution of one nitrogen base causes the inability of red blood cells to carry sufficient oxygen due to its shape. And then we can have insertion and deletion nitrogenous base mutations. So insertion, a base is added to a DNA sequence where ACG-CCA becomes AAC-GCCA, or deletion, where it would become CGC-CA. And in cystic fibrosis, we have the deletion of the 7th, 8th, and 9th nitrogenous bases, which then causes an inability to produce a protein that regulates chloride channels. Now, both insertion and deletion mutations now cause what we call a frame shift mutation. So because we've added a nucleotide, all the amino acids shift kind of one codon to the right, or one codon forward, or one codon backwards. And because of this, every amino acid after the point of mutation may be affected. So the insertion and deletion mutations typically are more serious than the replacement mutations. Translocation mutation is when a sequence of nitrogen bases is removed from one area and placed in another. So if we had A, B, C, D, E, F, G, H, I, J, then we had A, B, F, J, H, I, F, G, H, I, J, and then C, D, E. So we have the end placed now into the middle. We can also have an inversion mutation, where we have a reversal of a sequence of nitrogen bases. So A, B, C, D, E, F, G, H, I, J becomes A, B, EDC, and then FGHIJ. So a section of those nucleotides have been reversed. Again, now that might affect one or two amino acids, but it's not necessarily going to shift it down the whole chain. Duplication mutations end up duplicating a set or sequence of nitrogenous bases twice in one location. And luckily, there also is silent mutations. And now any types of these mutations could be silent if there's no phenotypic effect. So remember how we spoke about the redundancy of the genetic sequence, where we have lots of different mRNA codons that can produce the same amino acid. So if we have the mutation, point mutation GTA, and now we've gone GTG, we've changed our mRNA from CAU to CAC, and they both code for the amino acid histidine. So we wouldn't notice that this mutation occurred because there's actually no change in the amino acid. And luckily, the body can repair some mutations during that replication of DNA as the polymerases check for errors, but not all mutations. And some mutations can lead later on in life to developing cancers and other problems. I hope you enjoyed our lesson today on mutations. See you next time.